nutrient media at 37 degrees centigrade. The numbers get so large, we express them as the log of the number of cells. This is growth curve of bacteria. About growth curve of bacteria, I explain next session. But you know, the growth curve of bacteria include some stages: A, lag phase, B, exponential phase, C, maximum stationary phase, and D, death phase. I will explain it in next session. Microbial growth is affected by two major factors. One, environmental factors. Two, chemical factors. Environmental factors like temperature, pH, osmotic conditions. Environmental conditions such as temperature, pH, osmotic conditions, and chemical factors are proper concentration of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, some trace elements, and some organic cofactors. I will explain about chemical and environmental factors involved in microbial growth this session. Temperature and microbial growth. Temperature and microbial growth. Cardinal temperatures. Three temperatures are very important for us. Minimum temperature, optimum temperature, and maximum temperature. Minimum, optimum, and maximum temperature. The temperature is a major environmental factor controlling microbial growth. Each organism has a minimum temperature, optimum temperature, and maximum temperature that can grow. Minimum temperature, maximum temperature, and optimum temperature. In optimum temperature, enzymatic reactions occurring at maximal possible rate. Yes, it's okay. It's usual. In optimum temperature, it is the best temperature for that organism. It's the best temperature. In optimum temperature, enzymatic reactions occurring at maximal possible rates. And in minimum and maximum temperature, enzymatic reactions occurring at increasingly rapid rates. Yes? Membrane yelling, transport processes so slow that growth cannot occur in minimum temperature. Growth cannot occur because the growth is slow. And in maximum temperature, we have protein denaturation. Yes, in high temperature, some proteins, some enzymes degraded and denaturate. Denaturate of proteins, of enzymes occurs in maximum temperature. Collapse of cytoplasmic membrane and thermolysis. Temperature. 3 temperature: minimum, optimum, maximum. What's minimum temperature? Temperature below which growth decreases, or lowest temperature at which microbes will grow. Temperature below which growth decreases, or lowest temperature at which microbes will grow. Optimum temperature. It's the best temperature. Temperature at which its growth rate is the fastest. Yes, fastest state for a microorganism is in optimum temperature. Maximum temperature. Its temperature above which growth ceases, or highest temperature at which microbes will grow. Maximum temperature is temperature above which growth ceases, or highest temperature at which microbes will grow. Classification of microorganisms by temperature requirements. Yes, classification of microorganisms by temperature requirements. Psychrophile grows in cold condition. Psychrophile from 0 to 10 degrees centigrade. And the best degree centigrade is 4, refrigerator temperature. It is refrigerator temperature, 4 degrees centigrade, 0 to 10. At first, psychrophile, and mesophile, green, mesophile. Mesophile, for example, from 10 to 15. Optimum in mesophile is 35 to 37 or 39. After that, thermophile, psychrophile, mesophile, thermophile. Thermophile, like heat. For example, Bacillus stereothermophilus. Mesophile, such as Escherichia coli, Staphylococcus aureus. Psychrophile, such as Chloromonas, Pseudomonas, Acinetobacter, for example. Thermophile, the best time for these organisms is 60 centigrade. Then hyperthermophile, over that 16. The best temperature in this state is 88. For example, for Thermococcus, this is not bacteria, it is archaea. Yes, it is archaea. Thermococcus is an archaea. And other hyperthermophile, for example, Pyrolobus. Pyrolobus is archaea too. And the best temperature for it is 100. Excuse me, just a minute. Affects the growth of bacteria is pH, acidity, pH, acidity, pH and microbial growth. pH measure of hydrogen plus proton. Each organism has a pH range and a pH optimum, like temperature. Yes, like temperature, each organism has a temperature range and a temperature optimum. And for pH, each organism has a pH range and has pH optimum. Acidophiles means an organism likes or grows in acidophilic pH, in acidic conditions. Optimum pH range for these organisms is 1 to 4. It's very low pH, 1 to 4. And alkalophiles. In alkalophiles organisms, optimum pH is 8 to 11. 8 to 11 alkalophile. Acidophile like lactic acid bacteria, thiobacillus bacteria, thiobacillus thiooxidans, thiobacillus ferrooxidans, lactic acid bacteria, they are acidophile. Lactic acid bacteria, optimum pH is 4 to 7. For thiobacillus is 2 to 3, for example. For fungi is 4 to 6. Internal pH regulated by buffers. Yes, buffers are regulating pH. Internal pH regulated by buffers and near neutral adjusted with ion pumps. Yes, adjusted with ion pumps, alkaline pumps, acidic pumps. In the cell, adjust the pH near neutral, near 7. Human blood and tissues has pH 7. Yes, 7. It's neutral, not acidic, not alkalophilic. pH and microbial growth. The acidity or alkalinity of an environment can greatly affect microbial growth. Most organisms grow best between pH 6 and 8. But some organisms have evolved to grow best at low or high pH. Most organisms grow best between pH 6 and 8. It's about neutral pH. But some organisms have evolved to grow best at low, for example, thiobacillus, and high, for example, in Vibrio. The internal pH of cell must stay relatively close to neutral, even though the external pH is highly acidic or basic. Acidophiles. What's acidophiles? Organisms that grow best at low pH. Yes, organisms that grow best at low pH. For example, Helicobacter pylori, Thiobacillus thiooxidans, lactic acid bacteria, for example, Lactobacillus lactis, Lactobacillus cremoris, Lactobacillus bulgaricus in yogurt, and alkalophiles, organisms that grow best at high pH. For example, Vibrio cholera. Most of pathogenic bacteria are neutrophiles. Yes, because pH of our body is neutral, and most of pathogenic bacteria are neutrophiles. They grow in neutral conditions. Osmotic effects on microbial growth. I spoke about temperature and about pH. This is about osmotic effects. Osmotic pressure depends on the surrounding solute concentration and water availability. Yes, osmotic pressure depends on the surrounding solute concentration and water availability. Water availability, water presence, water availability is generally expressed in physical terms such as water activity, AW, water activity. Water activity means water availability in organisms. Water activity is the ratio of the vapor pressure of the air in the equilibrium with a substance or solution to the vapor pressure of pure water. This is formula about AW, about water activity. Pressure solution to pressure water. Pressure solution and pressure water. About osmotic pressure. About osmotic pressure, in different media, we have different form of organisms. For example, organisms which thrive in high solute means osmophiles. Yes, osmophiles is a definition for organisms that thrive in high solute. Osmotolerant. Osmophile means osmotic pressure like, but osmotolerant means osmotic pressure tolerant, can tolerate, not like. Osmotolerant, organisms which tolerate high solute, not like. Osmophile like, but osmotolerant means tolerate. Halophiles, organisms that thrive or that like in high salt means halophile. Some organisms in oceans are halophiles because of the high concentration of salt. And halotolerant organisms which tolerate high salt. Organisms that tolerate high salts means halotolerant. Borophiles, organisms which thrive in high temperature. Organisms that like in high temperature, that grows in high temperature, but barotolerant organisms which tolerate high temperature. You must know about osmophile, osmotolerant, halophile, halotolerant, barophile, barotolerant. Halophiles and related organisms. Halophiles and related organisms. In nature, osmotic effects of interest mainly in habitats with high salt environments that have reduced water availability. In nature, osmotic effects of our interest mainly in habitats with high salt environments that have reduced water availability. Halophiles have evolved to grow best at reduced water potential and some extreme halophiles, for example, halobacterium, Danielella, even require high levels of salts for growth. Halotolerant can tolerate some reduction in the water activity of their environment, but generally grow best in the absence of the added solute. Grow best in the absence of added solute. And xerophile will grow very dry environment. Water, low water is low water are able to grow dry environment. For example, Streptomyces bacteria are xerophile. It is dry, like powder. This curve, this shape is about halotolerant, extreme halophile. To chloride of sodium chloride, extreme, for example, staphylococcus, non-halophile, for example, Escherichia coli. Please remember these are very important for the Escherichia is non-halophile. And Staphylococcus is halotolerant. Vibrio fischeri is halophile. And halobacterium is extreme halophile. Do you have my voice? Yes, do you have my voice? Voice is not fine. Do you have my voice? Comedy, yes. It's about internet connection. Please disconnect and again. Please. And again. Please connect again. Microbial nutrition. Microbial nutrition. Why is nutrition important? Nutrition is important for all of organisms. It's for growth. Yes? Or nutrition is important for human. For human, it's very, very important, not? And for all organisms. Why? The hundreds of chemical compounds present inside a living cell are formed from nutrients. The hundreds of chemical compounds present inside a living cell are formed from nutrients. Macronutrients and micronutrients, macronutrients or micronutrients or micronutrients. Macronutrients, elements required in very large amount. Yes, macro. Macro means large amounts. Micronutrients or micronutrients, metals and organic materials needed in very small amounts, trace amounts, in very small amounts. Main macronutrients: carbon, 50% of dry weight, and nitrogen, 12% of dry weight. Autotroph. We divided the organisms because of carbon source to autotroph and heterotroph. Yes, autotroph because of the source of carbon. You can divide it, you can classify the organisms in autotroph and heterotroph. Autotroph are able to build all of their cellular organic molecules from carbon dioxide, CO2, carbon dioxide. Autotroph means they can build all of their cellular organic molecules from carbon dioxide. Nitrogen mainly incorporated in proteins, nucleic acids. Most bacteria can use ammonia, NH3, ammonia, and many can use nitrate, nitrate, NO3, nitrate. Nitrogen fixers can utilize atmospheric nitrogen, N2. Classification of organisms based on sources of carbon and energy used. Yes. Because of source of carbon, you can divide organisms in autotroph and heterotroph. And because of energy source, you can divide it to phototrophic, yes, or chemosynthetic, chemotroph, phototroph, or heterotroph. In this table, you can see this classification of organisms. Carbon sources, yes, carbon sources. Sometimes carbon source is carbon dioxide for autotroph, and for some bacteria and all of fungi, the source of carbon is organic compounds, organic material, yes. This is heterotrophic. This is heterotroph. And about energy source. When energy source is light, the organisms is phototroph. And when energy is chemical compound, organisms is chemotroph. Yes, phototroph, chemotrophs. What is phototroph? What are phototroph? Plants, algae, cyanobacteria. They are use H2O to reduce carbon dioxide, producing O2 as a byproduct. Photosynthetic green sulfur and purple sulfur bacteria don't use H2O, not produce O2. They use H2S instead of H2O. Photoheterotrophic. In photoheterotrophic, light source is energy source is light, and carbon source is organic compound. For example, green non-sulfur and purple non-sulfur bacteria. And about chemical compounds as energy source, these organisms are chemotrophs, such as hydrogen, sulfur, and nitrifying bacteria. And chemoheterotrophs. In chemoheterotrophs or heterotrophs, energy source is chemical compound, and carbon source is organic material, organic composition, or organic compounds, such as aerobic respiration. Most animals, fungi, protozoa, many bacteria, anaerobic respiration. Some animals, protozoa, bacteria, fermentation, some bacteria, and yeast. It means in chemoheterotrophs, we have three types of metabolism. Anaerobic respiration, aerobic respiration, and fermentation. Three forms of metabolism: aerobic respiration, anaerobic respiration, and fermentation. Other macronutrients. I said about carbon and nitrogen. Other micronutrients. What are other micronutrients? Phosphate, sulfur, potassium, magnesium, calcium, sodium, and iron. Yes, and iron. Iron plays a major role in cellular respiration, being a key component of cytochromes and iron sulfur proteins involved in electron transport. Then, iron is very important for respiration, cell respiration, for electron transport. Siderophores. Siderophores are some proteins, are some agents that chelate Fe. Siderophores means iron binding agents that cells produce to obtain iron from various insoluble minerals. Some bacteria can produce siderophores. Yes, some of pathogenic bacteria, for example, Mycobacterium tuberculosis. And some bacteria can produce siderophore. Because of producing siderophore, these bacteria can absorb Fe, iron, from plasma, from serum, in blood, and producing the siderophores is a virulence factor for some bacteria. Siderophores are iron binding agents that cells, means organisms, produce to obtain iron from various insoluble minerals. Another environmental factors, yes, I said about heat, about temperature, about osmotic pressure, about salt. This is about oxygen. Anaerobes lack superoxide dismutase and or catalase. Anaerobe organisms don't have the enzymes named superoxide dismutase and catalase. Anaerobes need the high, sometimes to remove oxygen. Chemical, thioglycolate, pyrogalol or pyrogalol, or NaOH, sodium hydroxide, H2 generator, plus catalyst. Physical, removal, replacement. This is a type of gas pack, a system for anaerobic culturing. When you want to culture an anaerobic bacteria, you must use this system. Yes, you must use this system, anaerobic jar or gas pack. And you have to use some chemicals to observe oxygen. For example, thioglycolate, pyrogalol. Special culture techniques. For example, candle jar. This is candle jar. Candle jar. This is candle. Sometimes you culture plates, tubes, and put it candle jar and put a candle and lid. When candle is turned off, carbon dioxide is formed and needs for culturing of the microorganism in tubes in your petri dish or plate, petri dishes or plate. This is candle jar. And gas pack. This is gas pack. Yes, for anaerobic growth. For anaerobic growth. Gas pack, CO2, N2 exists, and remove oxygen. Culture media composition. When you want to observe, to study an organism, most of the time you need culture it, yes, and follow you need culture media, suitable culture media. We have different culture media based on the compositions. I tell you about the different of culture media for organisms. Culture media supply the nutritional needs of microorganisms, carbon, nitrogen, phosphorus, trace elements, etc. Defined medium, defined medium, precise amounts of highly purified chemicals. Complex medium or undefined, highly nutritious substances. In clinical microbiology, we have selective media, differential media, basic media. Selective media means contains compounds that selectively inhibit. For example, SS media for Salmonella, Shigella. For isolation of Salmonella and Shigella, we can use a selective media named SS agar. Salmonella, Shigella agar. Yes. Differential media contains indicator. You can see, for example, lactose fermentation, glucose fermentation, H2S production. These media are differential. Terms that describe media used for isolation of particular species or for comparative studies of microorganisms. Terms that describe media used for isolation of particular species or for comparative studies of microorganisms. For example, a general differential media is EMB, eosine methylene blue agar. Gram-negative bacteria can grow in this media, but gram-positive not. And you can isolate gram-negative from gram-positive in this medium. Another example is McConkey media. McConkey EMB media are differential media. gram negative can grow and gram positive cannot grow in these media. Because of that, this media is differential. Types of media. Types of media. Media can be classified on 3 primary levels. 1. Physical state. 2. Chemical composition. And 3. Functional types. Physical state of media. Physical state. About appearance media. Some media are liquid, like water, liquid. For example, nutrient brass. Some media are semi-solid, between brass and solid, semi-solid. Some media are solid, can be converted into liquid. Solid cannot be converted into a liquid. Liquid media. Water-based solutions don't solidify at temperatures above freezing, tend to be free-flowing. Includes broths, milks, and infusions. Measure turbidity. Example, nutrient broth, methylene blue milk, thioglycolate broth. Yes, it's general media in broth form. We use liquid media for growth of bacteria, for turbidity of bacteria, of organisms. Semi-solid media exhibit a cloth-like consistency at ordinary room temperature. Cloth-like or gel-like or cloth-like, semi-solid, between brass and solid. Semi-solid media is used or are used for motility of bacteria. For example, SIM media. We have a media named SIM or motility media. This media is semi-solid and bacteria can move, and motility test. Used to localize a reaction at a specific site. Example, sulfide indole motility. Yes, SIM. S sulfide, I indole, M motility. Three tests in one media: sulfide production, indole production, and motility in this one media. For hydrogen sulfide production and indole reaction and motility test, 3 tests in one media. And solid media. Firm subsurface for discrete colony growth. When you want to see colonies of a bacteria, you have to use solid media. Yes, solid media have agar 1.2 to 1.5% agar. present in this media, solid media. Film surface for discrete colony growth, advantages for isolating and culturing. Two types. Yes, two types. Liquefiable, reversible, non-liquefiable. For example, gelatin and agar, liquefiable. Cooked meat media, potato slices, not liquefiable. Chemical composition of culture media. Don't tire, 10 minutes rest and then continue. A type of chemical composition. For example, one synthetic media, chemically defined, contain pure organic and inorganic compounds. Exact formula, little variation. 2. Complex or non-synthetic media contains at least one ingredient that is not chemically definable. Extracts from plants and animals. Yes, for example, sometimes serum of human, serum of rabbit added to this media. Non-exact formula tends to be general and grow a wide variety of organisms. Complex media can lead the growth of most of organisms because of specific and general and suitable composition in these media. Selective media. Selective media, for example, when Staphylococcus aureus grows in this media, this bacteria can ferment mannitol and produce acid, and then phenol red convert to red, from red to yellow, and yellow colonies is appeared. Media, yes, from pink or red to yellow because of mannitol fermentation in this media. Differential media. Differential shows up as visible changes or variation in colony size or color, in media color changes, or the formation of gas bubbles and precipitates. For example, EMB, eosine methylene blue. Eosine methylene blue is a differential media or McConkey. In this media, gram-negative bacteria can grow and gram-positive not because of salt, boil, some dye, for example, crystal violet, methylene blue, eosine. Example, sprit blue agar to detect the digestion of fats by lipase enzyme. Positive digestion, hydrolysis, is indicated by dark blue color that develops in the colonies. Blood agar for hemolysis, alpha, beta, and gamma hemolysis. EMB, McConkey agar, and other examples are differential media. Blood agar is a media with blood. When a bacteria hemolyzes the blood completely, the type of hemolysis is beta. When a bacteria hemolyzes blood incompletely, the type of hemolysis is alpha. And when bacteria couldn't hemolyze blood, this hemolysis is gamma. Alpha, not completely, beta completely hemolyzes, and gamma no hemolyzes. Oh, this is repeat. Enriched media. Enriched media for high nutrients, is used to encourage the growth of particular microorganisms in a mixed culture. Some of bacteria are fastidious, or they need many of ingredients, many of nutrients for growth. And we must grow these bacteria in enrichment media. For example, mannitol salt agar for Staphylococcus aureus, blood agar, chocolate agar, selenate F+, enrichment media. Bacterial colonies or solid media. This is Serratia marcescensis with red pigment in Serratia marcescens. Shigella flexneri in McConkey. Pseudomonas aeruginosa on TSA. Trypticase soy agar. Shigella flexneri in McConkey and Serratia marcescens, for example, in nutrient agar, in Muller-Hinton agar with red pigment. Laboratory cultures of microorganisms. Sometimes selective media contain antibiotics. Yes, is a question. Sometimes we add some antibiotics, for example, penicillins, to a media, and this media will be selective. Yes. For example, media name Campi BAP for Helicobacter pylori. This media have 3 antibiotics. Laboratory cultures of microorganisms. Microorganisms can be grown in the laboratory culture media containing the nutrients they require. Microorganisms can be grown in the laboratory in culture media containing the nutrients they require. Successful cultivation and maintenance of pure cultures of microorganisms can be done only if aseptic technique is practiced to prevent contamination by other microorganisms. Yes. successful cultivation and maintenance of pure culture, no contamination. Pure culture of organism can be done only if aseptic technique is practiced to prevent contamination by other microorganisms. You use laminar airflow when you want culture a bacteria, a fungi, to prevent contamination. This session is finished. Don't tire. Good afternoon. Thank you for your attention. And I'm ready. If you have any question, you can send me this session or via WhatsApp group. Thank you for your attention and for your times. And Saturday, you must tell me about another time of classes, because we have to finish the class in 2 weeks to next week, and we have to must teach at least 13 sessions, 13 lectures. And this today I teach 7, yes, 7 lecture, yes, 7 lecture, and I have to teach 13 lectures. Yes, thank you. Don't tire. Good luck.